This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 732, recorded on March 16, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, Vincent and everyone. And I'm looking out my window, as I usually do at this time on Tuesdays or Fridays, depending on when I come on. And last week, we had the most remarkably beautiful 2 days that harbingered spring that you could believe. And today it looks like winter again, and we actually saw snowflakes.
Dixon, I can see you through the arches of the bridge, right?
You can. I'll wave to you. Here, I'm waving to you.
Yeah, so I see the George Washington Bridge, and there's the arch of the far,
That's
what
right.
is it called, a pylon or something? And Dixon's
Stanchion?
Dixon's right in there. Isn't
Exactly.
that amazing? We couldn't have planned it better.
In fact, I did plan this.
Yeah, winter is back here. It's freezing today.
It is. It is freezing.
Also joining us from Madison, New Jersey, Brian Barker.
Hi. Unsurprisingly, it is also freezing here. Gray and 34 Fahrenheit.
From Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 45 Fahrenheit, which is 10 Celsius, partly cloudy, partly sunny.
And from Austin, Texas, Rich Condit.
Hi, everybody. 71 Fahrenheit, headed for 80. It's kind of gray, but I'm not going to complain after hearing your weather reports.
Yeah, it was -3 this morning on Wake Up.
Yeah, wait till you hear this. Here it comes.
Okay,
Yeah.
yes, we have two guests today, and I was going to say, and probably predict it, 72 and sunny there from San Diego State University's Forrest Rohwer. Welcome back to TWiV.
Hello, thank you. It was a little cold for us. We got down into the mid-60s.
And also from San Diego State, Maria-Isabel Rojas. Welcome, Maria-Isabel.
Hi, everyone. Thank you for having me. It's sunny and beautiful out here in California.
So, Forrest, I don't know if you remember, you were on TWiV 391 at Penn State.
Yeah.
It was quite a long time ago. And I think, Rich Condit, you were there with me and Alan Dove, and we also had David Price, who is also from SDSU, right? Yeah. That was a long time ago. It was a lot of fun. And because Alan was with us, the title is Whiter Reefs, Fresh Breath. Because David does oral virome, and Forrest talked about reefs and viruses. There you go. No reefs today, though.
My take-home message from that whole event was that mucus is cool.
Yes, that's what Forrest told us. Mucus is cool, right?
Yeah.
This is cool.
It's very cool. Okay, so today we have an interesting community project, which you're going to tell us about. But first, now, Forrest, you told us your history back at Penn State, but Maria-Isabel, can you give us your history before we talk about science?
So I am an Ecuadorian. I'm from South America. I've worked with infectious diseases mainly for my undergrad, and then I came to San Diego State for my master's and stayed for my PhD. I worked before COVID. I worked mostly with phages, especially focused on the lysogenic to lytic switch and its regulation everywhere in the human gut and a little bit in the ocean as well. And then as COVID hit, I transitioned to this community outreach and COVID in the environment project.
So, Forrest, have you abandoned phage and reefs for COVID-2 now?
Oh, God, no.
Good. I'm glad to hear it.
I was stuck at home, though.
Yeah.
So, hard to get to coral reefs at the moment.
Yes. Maria-Isabel, some time ago you sent me an email, which I never saw because there are too many there, but somehow Kathy and Rich rescued it or something. I don't know how it worked out.
I had nothing to do with this.
You
It's
had
all
nothing.
Kathy.
So Vincent, this original one that you pasted in the show notes, you read, and we talked about it on the show.
I did. Oh, okay.
Yes, so you don't even remember that.
No.
No,
Marisa,
Vincent
well-
remembers everything.
Yeah, right.
You will confirm that, right, Marisa? That
Give us a very kind shout out.
Yes.
We're right in the middle of recruiting all of these volunteers from San Diego County. So say thank you for that.
so I think it was sometime in June, maybe. And
Okay. So then we posted the link so people who were there could participate, right?
right. And I don't know how many others you got from TWiV, but I know you got one because my friend Sarah's daughter lives in San Diego. And so Sarah and I cooked up right away that Madeline should get one of these kits. And so then we actually got to see her do some of the sampling while we were on FaceTime with her. So I feel like I sort of participated from a distance. So that's
Okay.
so I don't know. Do you know? Well, you can tell us more later, but I'm just wondering if you know if you got others because of the TWiV bump.
Some people, so we shared the link of TWiV on social media as well. Social media helped us with at least that fraction of the users. And some of them, yeah, of course, some of the listeners too, right?
So that's why we're here today. Kathy arranged for Forrest and Maria-Isabel to come and tell us about this sampling project. So maybe you could start at the beginning. So you're a phage lab, you're a coral reef lab. What were you thinking?
So we got together that morning, the last morning that we all got sat together without masks and distance, but still, not knowing all of that, how long this was going to last. And the main question was that Forrest asked all of us was, what can we do to contribute to this effort? What can we contribute to this effort? A lot of people in the area were doing stuff with patient samples, right? And directly with the disease. And so we thought, and also, as you remember, the main thing that was put out there by the authorities was to clean everything and to be very careful about everything you touch, etc. And people were spending a lot of effort and a lot of money too into that sort of protection. So we thought, well, let's go out there and see if it's out there, if it's really out there, and how often do we encounter it. And while we're at it, not only we will engage the community, but we'll also look at what other things are out there, what other RNA viruses we are interacting with.
And it was working very closely with actually the UCSD crowd also. So there was like Rob Knight's lab was setting up to do a whole bunch of like in-hospital stuff. And then Kim Crawford was doing aerosol stuff and some water stuff. And so we figured we'd do surfaces. So we kind of get a good idea of what was going on in San Diego in general. So we were very tightly talking to everybody at that time.
It seems to me that in terms of expertise, your lab has a lot of experience and expertise in virus discovery and sequencing and that kind of stuff, which fits right into this, right?
Yeah, historically, the first time we ever did an RNA virome was actually for the SARS-1. So when SARS-1 came out, we'd just done what became Viromics, right? So about 2002 is when we published that data, and that was all DNA viruses. And then, so 2003-4 is when SARS-1 was going on. And there was a group from the Genomic Institute in Singapore that wanted to know how to do uncultured viruses and to figure out what was going on. And so they came over here, and that's when we started doing RNA viruses. Which was fun. I guess it's like almost 20 years later, you're running into yet another one of these nasty little viruses.
I love the term uncultured. I think it's perfect.
Uncouth.
So, Maria, so what did you do first? You had this idea So how did you implement it?
So the challenges were we want to collect thousands of samples. We want to collect them as unbiased as possible throughout this big area, which we delimited to be San Diego County. And San Diego County, as you know, has a big proportion of the population is a Spanish-speaking population. So these were our challenges, and the way we faced them was we created a very friendly call to action that we released in the media, the newspaper, the television, the social media, and to encourage people to participate. At the same time, we created this simple protocols with simple steps and a video on exactly how to sample. And the protocol was also graphic. So it was really easy for any age and any background to follow. We also created an online platform that served as a database for the lab, but also to interact with the citizen scientists. And for them we designed it in a way that it was very easy for them to click a couple buttons to upload the data of their sampling. And because we were very careful with social distancing and with being aware that a lot of people were staying at their homes and not leaving their homes, we also had a system to deliver the kits and pick them up from their doorstep. So we had a lot of users that were in complete lockdown because probably their medical situation or whatever, and we had others that were the complete opposite, moving around the city a lot and keeping their lives as usual because they had to work. So we were able to reach a broad range of the community, all ages, as I mentioned, and so that took care a little bit of how to get a large amount of samples throughout the San Diego County in a short period of time. And to tackle the constraint of the language, the platform and everything was fully bilingual, English and Spanish, and in some cases also with a little plugin for all the languages. And for this approach was very friendly for the early users, right? The people that are very keen to using technology. But we also try to reach the people that don't even send instant messages every day. So for some of them, we actually talk to them over the phone to answer their questions. We did a couple of small workshops on how to sample. They were outside in person, safely distanced, wearing N95 masks to actually demonstrate how to do it. And they would record their data on a piece of paper. So everything was completely analog. Their phones were not smartphones. So we were able to engage them too. And some of the community, it's worth mentioning that didn't want to disclose their home address, we would meet up at a safe point. We're at a public space where we can exchange the kit. So in that sense, we think we were able to reach a broad range of the community.
So did you have a specific type of services that you were asking people to be testing, or was it sort of whatever people were interested in, or how did the specific surfaces get chosen?
So any really, so people really want to help, right? People really want to know what's around them and what they interact with. And I myself was surprised to know how much people are interested in it. So we told them, first of all, don't go out of your safe zone. Don't do anything that would put you at risk or expose you more than you should in the time of the pandemic. But do swap surfaces that are of interest to you, to your lifestyle. And in some cases, there were people that were concerned, for example, about the homeless community in San Diego. So they swapped park benches, they swapped the crossing the street button. the public transport. Some people swapped the playgrounds because they wanted to take the kids out to the playground again, and they wanted to know if it was safe to do so. So there were all kinds of surfaces, cell phones. Some people swapped their church, the outside of their church.
So give us an idea of how many people in the lab were participating in this. How many people did you have going out and delivering the kits? And then also give us an idea of the numbers of people that participated, that got the kits and returned the kits and so forth.
Yeah, so initially we started with, of course, people in the lab. So that's when when everything was like late February, I guess, early March, when things are getting really ramped up. We were testing everything by using people in the lab. And then we reached out to the viral information group that's here. And so we got PIs in that group to do things. And then once we had it where we were happy with the kits, that's when we really reached out to the citizen scientists, like in a broader group.
So it looks like maybe about a dozen people are on each of these manuscripts or so. Is that right?
Yeah, exactly. And many of them were the people that were right there at the beginning, because we were, of course, running around, trying to get supplies, trying to get when we were, the test we actually used is the LAMP protocol as opposed to the reverse transcription PCR. Because one, we could get the reagents easier, and it's more robust when working with environmental samples. So that's kind of the route we went there.
I notice in the acknowledgments there's a Patricia Rohwer,
yes,
and a Gary Rohwer.
Yeah.
That can't be a coincidence.
Yeah, we sent him to Idaho and to Washington and to Madison because we had different people. We figured, if my parents could operate it, anybody could operate it.
Okay, good enough. Now I'm curious, because you said a lot of people had an interest in doing this because they were interested in finding out what was in their environment. Were you able to communicate the results of individuals back to them to tell them what their samples showed?
Yeah. So the system worked incredibly well. We thought we would be overwhelmed, and maybe internally we were a little, but in the end we were able to communicate with each one of them. Okay. We would send them updates. Of course, as you all imagine and now know, you've seen the manuscripts, most of the samples were negative, so we contacted them with good news. We gave them updates of the progress of the manuscript, but also we also reached out to them to ask them questions about when we were unsure about how they took the sample, or if they wanted to share any more details with us. And it worked really well. One of the people that is in our group is also bilingual, so he was also able to communicate with the Spanish-speaking community. And yeah, it was great. There was a lot of communication with a lot of them.
And we did the kind of the forensics follow-up on the positive samples. So we would contact the people directly whenever we found a positive, and then really figure out in more detail what was going on with that particular.
So the assay that you used was at least semi-quantitative, correct? I mean, did you got some quantitative feel out of it? Can you give me an idea of like what the record contamination was? Where did you find the most? And what level of contamination did you see?
So we ran over 3,000 samples we collected many more than that, but 3,200 samples had good data that we could work with, and from those 20 were positive. And we didn't see them clustered on any particular surface, meaning any type of surface, say wood, metal, etc. And we also didn't see them clustered on any particular area of interaction. Say, they weren't all in playgrounds. They weren't all in buses. They were really spread out through the sampling categories that we put all of our samples in. Yep.
And then the most positive one, how much did it have?
The most positive one probably had, it must have had over 1,000 viruses. We sequenced that sample and we were able to recover bits and pieces of the virus that were scattered throughout the genome of the virus. And remember, these samples are 10 by 10 centimeters surfaces that were swabbed onto a 1 centimeter square swab. So it's very, very little sample. So for us to have recovered that much of the SARS virus, that specific sample must have had a lot.
Right. Do you know what? Do you recall what that sample, what surface that came from?
I have to say, I'm pretty sure that one was for the handlebar of the bus.
Yeah.
Okay.
It was the bus.
It was the bus. the outer hand, the one that you grab onto.
Okay.
Unsurprisingly, because that surface is probably one of the most touched in the city.
Right.
So you said 1,000, but you certainly mean 1,000 copies, RNA copies, not viruses, right?
Yeah.
Correct. Yeah.
Okay.
Yeah, we definitely, in fact, everything we did, because, to be careful / just to preserve things, we had, when they were doing the swabbing, they were using these little like Swiffer pads, literally, which are really good at picking up things with just an SDS detergent on it. And so you would kind of clean off the surface. And that went into a tube with Trizol to preserve the RNA. So we only know about the RNA. We don't know anything about whether it was a virus that could be effective or anything about it.
So tell me a little bit about the kits. You gave people kits to swab with, right? What's in that?
So the kit, again, we were trying to make it not suffer the shortage supply, right? Mm-hmm. And also easy to use and friendly, essentially. So we provided them with a small cooler. The cooler had a couple of ice packs, 2 ice packs, one that they can put in their freezer while the other one was inside the cooler. And we told them to swap them every day. Then a bag that had inside a little box with the pre-labeled tubes that already had the tri-cylinder. And these tubes had a number, right? The code. And the code is the same code they will find on the website, on the app that is also friendly for the mobile device. So they could just click on that number and enter the data. Mm-hmm. There were the swabs, a little bag with swabs, a little toothpick for them to push the swab to the bottom of the tube. Mm-hmm. Gloves, many gloves, a pair of gloves for each sample. They were instructed to obviously change the gloves with every sample, and masks. So we got them N95 masks as well, along with the appropriate hazardous material sheet so they can be informed of the hazards of the trisol, and this graphic protocol I told you about that had the link also to the video for swabbing, and also our Twitter handle. We created a Twitter handle to spread it out a little more.
So you mentioned there's Trizol in there in the tube, which of course is phenol and guanidinium thiocyanate. And were you worried that someone was going to spill that on them, on their skin?
Absolutely. the main worry. So we were very careful to keep pointing out, Trizol is dangerous. Make sure to flip the tube down, because how it gets on the cap and you pop it. We didn't hear of any disasters, but yes, that was probably the main concern. Okay. The other one is we were like, they could just throw this stuff in their freezer, and the EH&S people didn't go for that. So that's why the little coolers with that.
I see.
Yeah, you have the methods paper is extraordinarily detailed. I really enjoyed that, and one of the impressive things is that by far the biggest expense was the coolers, followed by the masks. You know?
Yes, but we didn't want to buy just styrofoam ones, right?
Yeah.
That we have now are reusable for a project in the future.
Yeah. So the actually, so you got all the coolers back, right?
Yeah, most of them.
I was thinking it would be a bonus for the participants to get a free cooler out of the deal.
Yeah.
If you want to do this again, we will provide Microbe TV branded coolers. How's that?
If there's time to get them made in time.
I don't think anybody wants to do this thing ever again.
Okay.
What I thought was cool is that you used a delivery route planning software with a device, an app that went to the smartphone, so that the people who were delivering and picking up the kits could have the most efficient way to get there and back. Really smart thinking.
That was really fun because it was actually the so my wife, who you guys know probably, Anka Segal, who's also a phage biologist, and she's been working on an MFA. And so she said, we're like, we need to hire someone to help deliver these things. And she's like, oh, I know this perfect guy. So we have this poet writer who also every once in a while drives Lyft. And he basically organized the whole
cool.
The whole thing, like the delivery and everything. And it did work really well, actually. It was surprisingly good. We did deliveries a couple times, and it was pretty cool because you saw all these different parts of San Diego that you would never go to.
Right. So this is a great lesson for people who have little or no faith in public health, to get them involved in a project like this and see how much work it really is to collect samples and to analyze them and then to draw conclusions based on them. I think, if every community had this as an exercise, once a year everybody shows up, here's your swabs, go out and let's look for something, we would have a much safer country to live in. I can guarantee that we would. And we would need to spend a lot less time explaining why we do things also. So you've done a great job in clearing the air, so to speak. Did you alert the San Diego Department of Health that you were doing this?
No.
Do they know that you did this now?
You further this. I think citizen scientists are underestimated. It's underestimated.
Yeah, absolutely.
How much they want to help. And how much we could all benefit from their work.
No question.
Yeah, as a former resident of San Diego, I was fascinated by the map where you show where the positives were and then where all the samples were. And it looks like a huge collection of samples were in Mission Valley, right along I-8. But that's not where most of the positive samples were per se. So I just found that map really fun to look at.
Yeah.
What? Tell us about. So you get all these samples back in the lab. What was the processing? How did that work?
I have no idea.
So the sample journey was it started in a computer as an ID, then came through the lab, got tagged, then traveled in a car into the user's home. It was swabbed, put back into the cooler, came back to us, and it was put in a fridge until some of us cataloged it. We first had to put it into the system. Then it was brought to the BSL-2 freezer. And what I did was pull the samples. So in order for us to be able to go through thousands of samples in a very short period of time, at some point, we processed over 600 samples in a week, and a couple of 4 days, 600 samples. So in order to do it, we pulled the samples. They were already in TRISO, and then we did the old school crude RNA trisal extraction. We added one step to clean a little more of the inhibitors that work wonderfully. And then we saved the DNA and the protein for the future. And with the RNA, we ran 2 LAMP reactions. It was a multiplex LAMP to detect the envelope gene and the nucleocapsid gene. And we ran it in 2 reactions, one with just the sample and another one with a spiking control, because although we got rid of inhibitors as much as we could, with the environmental samples, there's always some inhibitor that will sneak in and inhibit the reaction. And so we ran them both to eliminate false negatives. And then we looked at it because it's a colorimetric assay, right? But one of the things with these environmental samples is that sometimes they change the color of the reaction, regardless. So we also run a lot of gels. And in order to do all of this, as you know, you can't open a lamp tube in the lab because it contaminates everything. So we are working in different rooms in different buildings. So we have a room just for the samples that is a very clean room, a BSL-2, a tighter BSL-2 where we are actually running the lamp and the extraction, and another room with the thermocyclers, and another room in a different building for the gel. So a lot of contamination control went into it.
So when you got a positive, was it always positive for both of the 2 probes or usually one?
Yeah, so it's the assay is multiplex. So we don't know if it was one or both. We did run the positives more than once just to confirm them.
Yeah. And then
I'm really impressed with LAMP assay. I mean, I've been aware of this, okay? But to see that figure you have of the tubes showing the sensitivity with, a number of genomes and whether or not that you get the colorimetric change is really striking, okay? That you can just mix this stuff together and stick it in a water bath and get that clear a distinction. It's really impressive.
Yeah, and we did have to hack it a little because the assay was built for, the assay as it is not multiplexed. It's meant for other things. It was designed to be multiplexed for a kit, a closed kit, company kit that was designed by NEB for humans. And what we did was sort of talk to them, read their papers, try this, try that, and sort of assemble the same thing for the environment. Okay. And it wasn't the first, the second, or the 5th iteration either, right? We went back to the rolling surface amplification. Remember this assay where you have a linear probe that sees the target, it circularizes, and then what you're amplifying is actually the probe, right? Not the target. So we played with a lot of molecular biology before we hacked into the lamp.
How long did it take to do all of the samples?
Sorry?
How long did it take to do all of the samples that you processed?
How much? Like a month?
A month?
I'd say month of real lab.
Okay.
Absolutely. Yeah. I mean, once you get it going, it works really well. It's all the troubleshooting up before.
Yeah. You said you also did some sequencing of the products. Is that right?
Mm-hmm.
Did you do all of them or just a few of them or what?
We sequenced a couple to see if we could recover enough RNA. Yeah. Enough RNA would also be viral RNA. And once we saw good signals there, we sequenced the 20 positives and we picked 20 samples that were alike but negative. So that were taken in a similar location and at the same material surface.
Now you also did some sequencing to see what else was in these samples, right? Some metagenomic sequencing. Tell me, what the rationale there was and what you discovered.
So we wanted to know what else is there, right? What else we interact with, and we found a lot of fecal-associated viruses. A lot of tomato, a lot of pepper model, right?
Yeah.
Yeah, the pepper mild model virus is one that I know Vincent and I show in our classes, and from Maya Breitbart's work, that's so abundant. But what you also found was this ryegrass model virus. Does that have to do with a lot of whiskey drinking? I mean, the model stuff, we think maybe has to do with a lot of salsa and stuff like that. But the ryegrass, I don't know. It was a new one for me.
You know that I don't know why we see that one in this particular place. The pepper PMMV, right? That one was actually the one that we found the first time we ran the RNA virions, and it was like, and it's infectious after it goes through. So people eat it. It's mostly in the pepper sauces. And it's infectious after it goes all the way through your digestive system.
Wow.
So it's a pretty cool little virus, actually.
Yeah.
Just like polio, same thing. It's infectious when it comes out. Did you find any phages?
Yeah, we did. We found a few. We found a couple of achromophages.
I thought you'd be more excited than that. Yeah, we found a few.
So these the plant viruses that you're finding, you're figuring are actually coming from the people and not environmental samples of one sort or another. Environmental contamination. Okay.
Yeah, they're definitely. We always find them really strong in fecal matter. It's like a really good signal of human fecal matter is actually the PMMV in particular.
Okay.
Always there. Yeah.
That's interesting.
Wow, shocking. People have bad hand hygiene, huh?
Yeah.
Go ahead.
Oh, so you mentioned that you had gotten some samples from, Madison and whatnot to have people test to see how well the kits were working in their hands. Did you have enough samples from different locations to have any idea of, San Diego versus another location? Going back to the beginning of the show and your excellent weather, did you see any differences or were you able to even look at differences between different locations?
No, we didn't have enough samples. We we were doing it, especially most of that was done at the beginning. So there was hardly any real, COVID was still just on the uprise. And so we don't see any positives in any of the non-San Diego samples. Yeah.
Back to the genomic sequencing. I'm wondering if you'll tell us about the one that was positive for SARS-CoV-2. I mean, you mentioned it a little bit earlier, but can you correlate that with where that one was sampled from? And just tell us more about that result.
Not really. So what we found in that sample was that it looked like it had more or less the same community than the rest of the samples. The only thing was that it had enough of the SARS-CoV-2 RNA that it took over a big part of that of those reads for the viral RNA. So you will see it, right? When you plot it, you will see a lot of it, but it's mostly because it looks like it took over the reads.
Yeah. And you should tell them about the package, because that one was fun.
Yeah, so because we were able to, we were communicating so well with the citizen scientists, and we interviewed each one of the people that had collected a positive. So there were, let me back up and say this, that some people collected more than one positive on the same day. So we called them to ask, could this have been, cross-contamination? And one of these people that we talked to that was also a great volunteer, because he kept asking for kids to monitor, the development of his household and his surroundings. But so he had a package delivered, 2 packages delivered to his home, and a magazine, and he did this swabbing where he swabbed the magazine, he swabbed the outside of one of the packages, the outside of the other package, and then the inside of each one of the packages. And we found the positive samples outside and inside of one of the packages and none other of the samples that he swabbed on that morning. And he's a scientist, so he took all the precautions. So that was interesting that Amazon package had some deep interactions there on the outside.
Now, these positives, were you able to find out if they were in any areas where people were infected, actually?
We asked that too to the citizen scientists, whether they had gotten tested or they were aware. And they all said that they had not been aware. Some of them had actually been tested with the PCR test every week. Some of them had been completely quarantined. And some of them, of course, had no idea really because they had never been tested. They didn't have symptoms.
Actually, for the most part, we did ask very detailed. Like, so we know that a lot of them that did find positive samples were never COVID positive by the PCR test and have not developed the antibodies. So it really suggests that they just weren't shedding the virus, that they actually were running into it in their sampling.
Yeah.
That would be our guess right now.
Yeah, and another one of the positives, one of the people that had many positives, and at least 6 positives inside of this person's house, it's a person that had been tested. We're sure that this person was never sick with COVID, but the profession of this person required they to go to other people's homes and move around the community.
Tell us again the number of positives over the number of samples, just to recap that.
So 20 positives and 3,284 samples.
Okay.
So we should be careful there a little because we're being kind of conservative. So like, for example, where like one person got the 6 positives at we just called out one.
One, yeah.
And so the frequency was about 0.6%.
Yep.
And that's not going to change. You know, statistically, that's about the relative abundance out there.
And I don't know if we mentioned this, but was there any particular kind of surface, not kind, but location? You mentioned bus handles. That was the most common positive. Is that correct?
That was the one positive that we believe had the most.
That's right. Okay.
But there were surprising things. Like I was expecting to find it a lot in the bottom, the crossing button, right? And we didn't. It wasn't particularly highlighted among the positives.
The biggest group is actually the benches and the play areas. That's where we saw the most positive percentage-wise.
I see.
Yeah.
It sounds like most of the surfaces you're mentioning are kind of like hard metal surfaces almost. Did you think about different types of materials like, you know, people's clothes or other types of surfaces, and were there differences in positives there?
Yeah, nothing that jumped out, because there were, of course, a lot of samples from packages, food delivery, things like that. And nothing there's no real order to it as far as it looks pretty much random as far as I can tell. Yeah.
When you mention playgrounds and park benches and stuff, I'm thinking little kids with runny noses, you know, wiping their faces and smearing the snot all over the place. Yeah. You know?
Or
the mucus.
Mucus, there you go.
Or fecal contamination, man. Dirty hands.
True.
Yeah.
Right?
That's right. That's right.
So, Maria, scientifically, aside from the whole, you know, getting this together and the organization and so forth, scientifically, what did you learn from this? What's the bottom line at the end of this study? What should we know?
So we don't know anything. We know that the virus is out there, but it is not that common on surfaces.
Okay.
We know with full certainty that it is airborne, that we sure wear a good mask, and that hand hygiene is as important as it is for any other diseases, right? We should wash our hands. We should not touch our faces after touching common areas, but as a common healthy practice.
So our modeling, one of our colleagues did a lot of modeling based on this data. Mm-hmm. And it really suggests that the surfaces are not that important for this pandemic. I mean, it is there, but if they're contributing to the spread of the virus, it's really a very small percentage of the cases, honestly. Yeah. And there was, I'm not going to be too vicious here, but I think that that misinformation at the beginning really did make a big difference, because convincing everybody that it was just going to be on surfaces and not to worry about it as long as you didn't touch your face was a really bad message to get out for a virus that's clearly a respiratory virus. Right? Yeah, I know that sounds obvious, but it's actually true, right?
Yep.
It really
sure.
So during the time when you were collecting the samples, what was the rate of infection in the population? How did that match up with what was really happening?
So we sampled throughout, let's say, the very beginning. There's a period there where, interestingly, the county doesn't have any data, but we do. we have samples before
they
the
do
county.
Now.
And so that was, say, the pickup of the first wave, and then through the first wave, and then coming down of that first wave. And we didn't see strong correlations with the positive, but also bear in mind that it's only 20 positives. It's very few.
Yeah,
that's true.
It's hard to draw a correlation, but there wasn't any from our data. There was no correlation between the number of cases per zip code even, or number of new cases that on the day of the sampling on that zip code and the amount of positives.
Right. Because you see the way it's played out over a year, and every holiday season or every spring break or every winter break, there's the 2-week lag, and all of a sudden it just roars back. And it would be really interesting to see how valid your data are when you're looking at the same surfaces in a city like San Diego, which I think is a destination for kids where you can get away from the winter blues and et cetera. As to the periodicity of appearance and disappearance, how often does the San Diego Transit Authority wash down their buses, for instance? How recent was that on the handle? You don't know, right? Because you have no data. So it would be very interesting to see whether what they're doing has any effect at all.
Yeah, it would be interesting to see the bus. It was. Interestingly, right before they started cleaning the buses.
And now they wipe them down with Lysol, I bet.
One of your manuscripts that we got earlier has the title has changed, but it was "Surfaces rarely sanitized" or something like that. Was the part of the title of the paper, right?
Yeah.
Commonly touched, but rarely disinfected.
There you go. That title was changed apparently.
Yeah, we're definitely targeting initially the ATM buttons and the handles of that. That's where we thought we might actually find it. But honestly, the citizen scientists did a really good job of randomly giving us a whole bunch of samples and places. We did find it in places that I really didn't expect to see it, right? I didn't expect it to show up, like on the food packages, for example, right? I thought they would just not be there, but it was there in a couple cases,
So one of your papers is, as I've said before, a really detailed description of exactly how you carried this out, not only in terms of the methods for processing samples, but all the collection and categorization and et cetera. I'm interested in whether you personally or the scientific community in general, what do you see as the future of this kind of effort? Do you see this applied in different circumstances? You've worked out a lot of bugs that could be useful in application of this sort of approach in the future.
Yes, I think we tried to solve the problem, and we designed something for the problem, and the solution came with it. And I think as you say, the protocol is very detailed, and it will allow anyone in the future to establish a system including the back end, right? Because we put details from the back end of like how to build the platform, the online platform, through the front end with the citizen, right? By using also these platforms that people are familiar with. People are familiar with their emails. People are familiar with their cell phones, or with talking on the phone and recording things down. And then to the lab. That labs, at some point we talked about what if we need this to be done in a country where or in a city that doesn't have a lab that is fully equipped to run qPCR, right? And so it can be run in places where there is basic laboratory conditions, and with the shortage of supplies that we ourselves faced when we were doing the troubleshooting.
And it also gives you a background of what the city looks like, because, we've had some pretty bad viral outbreaks here, especially of fecal-borne, like hepatitis sort of outbreaks. And so now we do have something to work against, which I think would be quite, we could deploy it either through the citizen science route or with just a whole bunch of undergrads, right? Which we didn't actually talk about, but we did do that. We used all the undergrads that could not take labs to do a whole bunch of sampling. So we have several, 500 samples maybe to get through from one time period where all the undergrads in like the Bio 200 did the sampling too. So things like that are a good way of getting a big snapshot of the city too.
So
I'm wondering.
Sorry, go ahead, Kathy.
I was just wondering who you got to cut up all the Swiffer squares, and then did they come with the 0.6% SDS, or you had to add that?
We made everything from scratch. It was, the higher the constraint, the better the design. So
sure, that's true. That's true.
It was done from scratch. We cut it ourselves. We bought it at some point. We ourselves bought it from the store. We prepare the hand. So we also offer, I forgot to say, we offer hand sanitizer in the kit. We make the hand sanitizer at the lab because there was no hand sanitizer in the city. So we made everything from scratch, except for the trisol and the lamp reagent.
And right. By we remember, that means everybody else. I just showed up and they kind of all Swiffer things.
So I have questions kind of similar to what Rich had asked about kind of the future of this and, how you can see using some of this in the future. But also, Maria-Isabel, you're a grad student, is that right?
Yes.
And so how do you think that this impacts what you might, or does it, you could totally say it doesn't, impact what you might do kind of in the future or what things you're thinking about doing long term?
I think the main thing that I learned is that there's no such a thing as impossible, right? We did something from scratch with nothing in our hands. And it's been also a challenge for me to have to talk across disciplines. So I've had to work with a developer and who is working on the back end and the front end, but at that point, I'm not really a user. But I'm also not on his field. So I've had to communicate with him and establish something that is good for the user between the two of us. I've had to also talk to the graphic designer, right, to make the protocol. And now with the mathematicians to make the model. So I've had to learn a lot of things. And I think I've learned to incorporate all those disciplines into solving a problem that I wouldn't have otherwise thought of solving it through those disciplines. And also the thing of empowering other people. We empower the citizens, of course, but we also empowered members of the team to help us and to make a big difference, like Rafa Baron, who's the person that Forrest was talking about, that's a writer, and now he wants to be a scientist as well. And I think those skills are going to be very useful and important for my future.
What is your thesis work on, Maria?
All about viruses, essentially. So it's the two phases of viruses, right? How viruses can sometimes help the host, sometimes help themselves. A part of it starts with the beginning of it is about how prophages carry genes in pathogens versus non-pathogens, host-associated pathogens versus non-pathogens in the ocean. And we found more prophage-associated genes in the pathogens that are carbohydrate metabolism, nitrogen metabolism, and not surprisingly, virulence factors. Then I dive a little bit into the switch. So what is regulating the switch to go from lysogeny to lysis? And for this, I work with the stringent response. I don't know, some of you remember PPGPP?
Magic spot.
Yep. Sure do.
Magic spot goes back, what, 60 years?
Yeah.
Yep.
So we collaborate with Anka Segal's lab and Jeffrey Gordon's lab with both in Salmonella and in a Bacteroides strain to figure out how the switch is being triggered for the phage to escape the host due to the stringent response. And then I transitioned to the human viruses, a little about the what RNA viruses are out there in our environment and what we're interacting with.
So you do some field work, it sounds like.
Some field work.
Good. Love it. What are you going to do next when you're done with your PhD? Do you know?
So I'm hopefully going to be done in the summer. Let it be record of that. And I'm looking for a postdoc. I'm looking for a postdoc. I would like to keep working on phages or on viruses, mainly in how they interact with the host. And I would like to do more translational stuff, dive a little bit more into the translational stuff.
Well, I presume you'll have a chapter of your thesis devoted to this project. And it'll be an interesting chapter for sure. All right, guys. From San Diego State University, Forrest Rohwer. Thanks for joining us, Forrest.
Thank you.
Maria-Isabel Rojas, thank you. And lovely project. Great to hear about it.
Pleasure.
Thank you, guys.
Thanks. Great talking to you.
Thanks, Marisa. Thanks, Forrest.
Thanks. This was really cool.
Good luck.
Yeah,
Bye-bye.
absolutely.
Thank you. I just wanna mention that you guys were a big part of kept me company throughout all of those long lab hours. Yes.
Glad to hear it.
So you want some more jokes?
Good. Thank you again. Bye-bye.
Great.
Bye.
Bye.
A couple of weeks, we're going to have a similar project here in New York City. A bunch of academics decided to check the sewage for viral RNA. So we'll hear about that. And I want to announce a meeting. Barend Jan Bosch and some other coronavirologists from the Netherlands are organizing this year's online NIDO 2021 symposium. NIDO, Nido Virales, or NIDO. They're cool. This was a meeting planned for 2020, but that was canceled. I was invited to actually go and do a TWiV, but out the window. Damned viruses. This one is going to be cool. It'll be online, and you can go take a look at the scientific program. I will put the link in the show notes, and you can bet there'll be a lot of SARS-CoV-2. So the keynote speaker is going to be George Gao, Director General of Chinese CDC. Structure, virus entry, replication, etc., pathogenesis, a lot of good stuff. So check it out. You have to register. Let's see. 95 euros. So you can't just go, unfortunately. But if you're interested, check it out. All right, let's do some email to finish out today. And the first comes from Charles, who wrote, as you look through NPR this morning at the Peter Daszak article, and I wanted to just chat about this briefly because it's quite interesting. The title is, this is from Goats and Soda by Michaeline Duclef. You may remember, she was on stage with me at ASV a couple of years ago. WHO points to wildlife farms in southern China as likely source of pandemic. Now, in this article, there are no data which supports that. But hopefully we'll see. In 2 weeks, they're going to issue this report, and we will see what they say. But apparently so Peter Daszak is quoted extensively here, and he said the WHO team found new evidence that these wildlife farms were supplying vendors at the Huanan Seafood Wholesale Market in Wuhan with animals. It was shut down in February. Now, they've been saying it wasn't the Huanan Seafood Market. And so now I don't know what's going on here, but he says they have exotic animals like civets, porcupines, pangolins, raccoon dogs, bamboo rats. They breed them and they ship them. And, Peter talked all about that. And the thing is, February 24th, so, China shut down this farm. So they had originally promoted wildlife farming to help rural populations make a living, right? And he says in 2016 they had 14 million people employed in wildlife farms, $70 billion industry. Wow. And then on February 24th, 2020, they shut down the farm. They said they were stopping the farming. And he said that's because he thinks that's probably because this was the source, one of these farms. And he said they sent instructions out to farmers to how to safely dispose of the animals, bury or kill them or burn them in a way that didn't spread disease. Why would the government do this? Because Daszak thinks these farms could be the spot of spillover. So no evidence, just hearsay. So we'll see what happens. I suspect I doubt they're going to have any data. But anyway, I emailed Daszak today and I said, hey, when this report comes out, want to come back on TWiV? We're really nice to you, You'd like to come. So it will be interesting to hear from him. Okay, anyway, that wasn't the point of Charles's email. The second point was this other article, also in NPR. Scientists find a new invasive mosquito in Florida.
Yes.
Aedes scapularis. Mm-hmm. What does it, bite your shoulder blade? Just a joke, folks. You don't have to write email. Anyway, apparently has come from elsewhere and they've just detected it. I think this, University of Florida, although I don't know which campus, Rich, they're a bunch, right?
Yeah, lots of times when they say University of Florida, the Gainesville campus is pretty sort of arrogant about this whole thing. When you say University of Florida, the implication is that it's the Gainesville campus, and the others have other names. But I don't know that applies in this particular case.
Anyway,
another thing about this mosquito is that it can carry yellow fever.
Oh yeah, well, all Aedes aegypti, all Aedes can carry yellow fever, so that's kind of a generic thing. Okay. I think it's called scapularis because the back of it looks a little bit like Ixodes scapularis, which is a kind of a silvery coating on the back. So I think that has some Latinized meaning rather than scapula.
Dixon, they say scapularis is a mosquito that likes going indoors and feeds on both birds and people.
Yummy. Sounds like a West Nile virus.
Yeah, it's a culicine, right?
No. It's culicin. Of course it's culicin. Of course it's a culicin.
But they bite people. They bite birds and people, right?
Oh, yeah.
But they don't go inside necessarily, right? Culicines?
No, they do. The house mosquito. Your Culex pipiens is called the house mosquito.
Okay.
Yeah, this is University of Florida, Gainesville. This is the Institute of Food and Agricultural Sciences, IFAS.
Very good.
Right.
I presume you don't know the guy.
No.
Okay, doesn't matter.
Well,
Hey,
they have a
anyway,
great
Charles said,
entomology
normally I
program.
would say enjoy the read, but I cannot enjoy reading about a new mosquito in the U.S.
Yeah,
Sounds like
right.
Alan Dove. He doesn't like mosquitoes, does he?
Sorry, nobody. Well, I don't know. I guess I do know a few people that do.
Dixon, can you take the next one?
Certainly. Robin writes, hi. You read a letter from Josh on Sunday's TWiV that eviscerated California's vaccine procedure. That was not my experience at all. I'm a school librarian who has been working with students since October in the Learning Hub, so I've been exposed all year. No infection, tested twice so far. My school sent me a notice to get a vaccine through the My Turn website. I registered within 10 minutes, had my first vaccine 2 days later. I received my second vaccine last week. They sent me a reminder to get my second vaccine through my email and via text message, which was great. I went to the drive-through vaccination site at the Oakland Coliseum, and I have to say this was one of the best experiences I've ever had getting a vaccine, faster than getting my flu vaccine at Rite Aid. It was quick, extremely well organized, and I must say the FEMA people were wonderful. My husband is not eligible yet, but I put him on the list. Great work being done made me hopeful. It seems like Josh is just pushing a political agenda. The California government is incompetent. Stay healthy, Robin.
Yeah, we got a few emails saying that Josh's view is not the only view, so who knows, right?
Right.
I just felt badly that he lost faith in the whole system, right? Which we don't want to do. Brienne, can you take the next one?
Sure. Lisa writes, hello, TWiV crew. As far as I know, bats and U.S. slaughterhouses have few direct connections, but as just a veterinarian who has worked with both entities at different stages of my career so far, I have a few comments. Many episodes ago, I believe around 600, but apologies for my incredibly late response, there was a brief discussion about bats' modulated inflammatory response due to their high metabolic rate based on the physiological demands of flight. This led to pondering if bats had greater immune responses during hibernation or torpor. They don't. Along with all other metabolic processes, the immune response is essentially zero during hibernation. This minimal response is a reason why hibernating species of North American bats have been ravaged by Pseudogymnoascus destructans, the causative agent of white nose syndrome. As my supervisor described to me when I began WNS white nose syndrome research with him, the bats are just little bags of meat hanging in a refrigerated cave. P. destructans is a fairly wimpy fungus that can be readily cleared by a fully functioning immune system or, on surfaces, killed by many disinfectants. However, when bats rouse and begin to react to the infection, they will often have an overexuberant response similar to that of AIDS patients who have a sudden increase in immune function following treatment. So even bats can experience cytokine storms. And she gives a link. Although I am not aware of any evidence for infection of North American bats with beta coronaviruses, a number of state wildlife agencies were issuing orders preventing the rehabilitation of native bats lest an infected human pass on the virus to a bat that could later be released and create a wildlife reservoir in North America. This would have the inadvertent effect of further endangering the public when untrained people come into contact with a sick bat and they handle the bat because the trained and rabies vaccinated people can't assist. And she gives another link about public letters to help bats. I have lots of opinions about working and inspecting the slaughterhouses during this time, but I don't think my supervisors would appreciate me sharing those. Thank you so much for being a continuing light in this dark time. From Lisa, who is a DVM in Eugene, Oregon, where it is partly cloudy, 51 Fahrenheit, 10 Celsius. And she has a PS. I showed up the picture of the chicken breast in TWiP 185 to some of my very experienced colleagues. We all agreed that the worm, in quotes, was just likely connective tissue.
Remember that, Dixon?
Yes, I do. I do very distinctly remember that.
Yeah.
And I agree with them. I didn't think it was worm either.
Really?
Yeah, I didn't think so.
Okay.
Name which worm it would be in chicken breast. That's the problem. There aren't any.
There aren't any?
None that I've never heard of one even.
Well, I suppose you could just look at it under a scope and see that it's tissue, not a parasite.
Exactly right.
Yeah.
Exactly right. Exactly
Somebody
right.
sent us this. I remember.
Right.
Okay.
Brianne, I have to know, did you practice the name of this pathogen? Because
I did not actually.
well, that's impressive.
Rolled off your tongue, so to speak.
Can you tell I've been teaching emerging infectious disease earlier today?
Okay.
There you go.
And I would like to thank Lisa for this link about bats and cytokine storms, two things that I find really interesting. So I'm excited to read it.
Yeah.
Kathy.
Kermit writes, Dear TWiV, I'm a retired book publisher and editor who retired from publishing to go to physician assistant school several years ago. I grew up in Montana, did 20 years of penance in New York City, and now live in Vermont, where it's a beautiful sunny spring day with temperatures pushing towards 60. Episode 729 has a brief mention of the term apoptosis, with discussion of how many different pronunciations of the term are used. Unfortunately, none of those examples mentioned were correct. I just so happened to keep the original article proposing the term on my desktop, and he gives a link. And I read through the whole letter earlier and thought it was going to also give the pronunciation, but it only gives the origin proposing the term. Okay. Interestingly, the article bothers to include a footnote on page 241. Oh, I missed that, explaining the pronunciation of the term. The word apoptosis, and then there's this strange set of colons and letters, is used in Greek to describe the dropping off or falling off of petals from flowers or leaves from trees. To show the derivation clearly, we propose that the stress should be on the penultimate syllable, the second half of the word being pronounced like ptosis with the P silent, which comes from the same root, to fall, and is already used to describe drooping of the upper eyelid. In short, it's pronounced apoptosis with the accent on the middle syllable. So apoptosis. This is a pronunciation I've never heard used by an instructor or a medical professional. I'm hoping you'll like this insofar as no one seems to have this term right. Keep up your good work. I've been a fan of TWiV since I did Vincent's online Coursera course in one of its early iterations. Those Coursera courses inspired me to retire and go to PA school. Regards, Kermit.
Okay, so how do you pronounce this, Dixon? I'm curious.
Apoptosis. That's the way I used to pronounce it.
Okay, what about you, Brianne?
Apoptosis.
Rich?
I was told that the PT is the same PT in pterodactyl.
I'm pretty versatile,
Yeah.
but I think probably I'm more likely than not to say apoptosis.
Apoptosis.
Kathy?
And I'm likely to say apoptosis.
So Kermit is saying the correct way is apoptosis, right?
Apoptosis.
So we're all wrong.
Well,
Right.
and it talks about a middle syllable, and he spelled it out, or Kermit, I don't know if it's a he or she, spelled it out APPTOESIS.
Yeah.
And if you say that, when you get to the P and the T for apoptosis, it ends up being 4 syllables. So it's a good thing that he spelled it out, that Kermit spelled it out, so that we know it's 3 syllables. And there's probably a linguistic term for that, of the P of ap next to the T of toe.
So, and that's the pronunciation. Do I have this straight? That's the pronunciation proposed by the people who actually proposed using the term
Yeah.
Yeah.
in biology. So that should be the correct pronunciation, right? Because I've heard, really enraged arguments.
Really?
Right.
At like even thesis committee meetings
Oh, dear.
about how to pronounce this. All wrong. I've never heard this pronunciation before.
Apoptosis. Okay.
Apoptosis.
Apoptosis. I'm not sure I'll remember it, but I'll try. Apoptosis.
We'll remind you, Vincent.
You need to remember that it's a very apt pronunciation.
Apto. I'll remember the toe part. Toe.
Right.
Apoptosis. All right. Rich, can you take the next one?
Hang on. I went up and I was playing with titles. Thinking that there ought to be something about apoptosis. Jerry writes, Dear TWiV, first, thank you for your fabulous podcast and keeping us, meaning the world, informed about all things SARS-CoV-2 and COVID-19. This one is for Brianne, since she mentioned the Hamilton song on TWiV 726, and for Vincent, who said he thought this was the best song in the musical. The HERO Registry is an NIH-funded prospective registry of healthcare workers who have received the COVID-19 vaccine. As you can appreciate better than most, communication is challenging, but oh so important. Members of the HERO research team have put together this parody of the Hamilton song that you like so much. Full disclosure, which while I am not one of the HERO investigators, I'm in the registry. Enjoy, and gives a link. And this is from Jerry, who's an MD and NPH, and a professor of radiology and neurological surgery, and a bunch of other stuff at University of Washington School of Medicine. And I watched this video, and it's terrific. It's a really nice.
So good.
It is very good. Yes.
Very good.
Completely agree.
One more round. Colleen writes, Dear TWiV, I would have never thought I would have traded in watching nail art and hair tutorials to listen to a podcast on virology. But here we are. This is better. This is better. You have sparked an interest in all things virology, immunology, and science that I never knew was in me. I've never been a bookworm or very good at school, really. But after the pandemic hit, I got sick in March, and no one knew why I continued to be sick for so long. I found groups online. It was great to know there were others like me, but I needed to understand this virus, or at least listen to people that were breaking it down and discussing the facts, and that has been you all. I listen to every single one of your podcasts since I found you, and I anxiously await each one. I learned and understand terms I never thought I'd be able to pronounce, and even became the person friends came to help decipher information. Trying to understand how this virus worked and following the science on what was developing as it was happening helped me feel empowered and motivated me to forge ahead no matter how dire things seemed when I never thought I would get better. You helped me understand and feel confident in the vaccines and able to encourage friends and family who were on the fence. And even better, listening to Dr. Griffin, who I jokingly refer to as my doctor husband, to my actual husband. I'm not sure, but I'm sure he's fine with it. I felt confident in getting the vaccine even after there was no clear consensus on what to expect, since I am a long hauler with a history of multi-system inflammatory reactions, Kawasaki disease, then the full-on assault COVID attempted. All I heard was never miss a chance to vaccinate in Dr. Griffin's voice in my head. I received my first Pfizer 3621 a week before my 1-year COVID-versary. Thank you for sparking this interest in me. I already have some classes I'm ready to enroll in as soon as I'm done with my hopefully successful cognitive rehab with the Mount Sinai Post-COVID Clinic. That's right here in New York. Thank you for your trusted science, facts, and not only education, but comfort. Also, I really hope the National Bobblehead Company listens to my suggestions to make bobbleheads of your crew to put next to my Dr. Fauci bobblehead. Colleen. Great.
That's wonderful.
It's a nice fan mail. Appreciate it. So if you folks remember our long COVID episode, Fiona Lowenstein talked a lot about the Sinai long COVID clinic. It's really good. Dixon, can you take the next one?
I will. John and Ruth write, Dear TWivers, we are supporters of TWiV and enjoy even the arcane discussions. The clinical updates are a much appreciated weekly summary too. The question we'd like to present is, could you comment on a possible timeline for evaluation of molnupiravir?
Almost.
Piravir.
Very good.
Molnupiravir. Okay, fine. The summary below referenced this abstract, which is there, and I guess you're familiar with it, Vincent, because you know how it's pronounced, and it's summarized below in another post, and then John and Ruth. So I haven't got a clue. So please help me out here.
A smile is contagious too.
Oh, you're going to go
Asbestos.
further. Okay.
Asbestos.
That's right. Asbestos. That's right.
This was actually mentioned multiple times over the past year. First by, I can't remember his name, Vanderbilt,
Right.
antiviral guy at Vanderbilt.
Mark Dennison.
Mark Dennison. And
Mark
then
Dennison.
Lisa Berlinski mentioned it. It goes by other names. Then we did a paper about how the Mark Peoples, is that his name, at UGA? I think so.
Mark. I'm thinking of somebody at Ohio State.
No, yeah, that's Mark Peoples at Ohio State. Who's the guy at UGA? Anyway, from his lab, they showed it blocks transmission in ferrets, and now it's in a phase 2A. And Merck is partnering with Ridgeback Biotherapeutics, phase 2A results, where they enrolled people. And the primary objective was, and Kathy has transcribed.
I didn't transcribe. I just got it directly from Daniel.
Is that from Daniel?
Because I recognized it and I thought, didn't you just talk about this? And so I thought he would just write back and say yes. But instead he put in the whole transcript. Of
Okay, because he has notes for his thing. All right, so these are Daniel's notes. So the primary efficacy objective was reduction in time to viral negativity measured by RT-PCR analysis of nasopharyngeal swabs. This is an oral ribonucleoside analog, okay? And the secondary endpoint was reduction in time to negativity of virus isolation in cell culture. And they report results for 72 participants, day 5, reduction in positive viral culture in subjects who received all doses compared to placebo. 0% of those that received molnupiravir were positive by culture at day 5, 24% placebo.
Wow.
So that's cool. And this is an oral medication. So I think this is very promising, but it's got to do a phase 3 now, right?
Right.
Seems that way.
But that's what you need is an oral drug so that people who are candidates can get it easily.
Yep.
Exactly.
Brianne.
Sure. Karen writes, Dear TWiV, one of our biology teachers here at the high school where I am librarian recommended TWiV to me many months ago, and I have been an avid follower and fan ever since. As a layperson with just enough Google skills and a big enough lack of science background, I find myself going down rabbit holes all the time. My question, if one has been infected with SARS-CoV-2 and is afforded the opportunity to get a vaccine, should said person get it as soon as possible or wait until immunity wanes to get more bang for the buck, so to speak? I understand the public health response is to get vaccinated ASAP. However, given that seropositive individuals have greater reactogenicity after a single vaccine shot than a seronegative person after a single vaccine shot, I wonder if this person would have greater reactogenicity getting vaccinated on the heels of the infection versus, say, 3 months later. Or does natural immunity grow over the course of 3 months so that reactogenicity would be greater 60 to 90 days after initial infection? With the vaccine, antibodies increase over time, right? In our area, supply is not an issue anymore, so taking someone else's place in line isn't a concern. Is there any suggestion that this question even matters? Can you help me get out of this rabbit hole? Thank you for being a rock in this storm. Best, Karen, who is in the library media in Knox County Schools.
What would Daniel say? Don't pass up your vaccine?
Yeah.
Yes,
I'd like to know where there's no shortage. That's great, right?
Yeah, that's fabulous.
Many parts of the country, there are shortages, you know. So
I'm just looking up where Knox County schools are, and I'm thinking Tennessee, Knoxville. Yep.
could be.
So I guess maybe this is just a problem for me reading aloud, that I got confused in thinking about Karen's question, but I think I see it now, that Karen wants to know if this is going to change her immune response. For a second, I wasn't sure if she was worried about making sure her immune response was best or whether she was worrying about avoiding side effects. But I think she's trying to get the best immune response. I think you get vaccinated when you have the chance to get vaccinated.
Mm-hmm. Agreed.
Everything else is just stories.
Yep.
Right.
All right, Kathy.
Scott writes, TWiV team, greetings from northern Wisconsin. I'm just a high school shop teacher. Just a quick thanks for all the information you generously provide weekly. I found your podcast in April of 2020 and have been a listener ever since. I've gone on to listen to a number of the YouTube virology lectures from Vincent to get a better understanding of both the subject matter and the banter. With your help, I felt better as to how to prepare myself when we open school as normal in September. We have been 4 days a week, Monday through Thursday, in person, about as normal as before the pandemic, distancing where we could and doing some sanitizing at least once a day with electrostatic sprays. For background, Friday is our day to prepare and record lessons for the following week, as we are not doing synchronous at-home learning. Our online students struggle with internet availability. Our building is 1981 construction, and the air handlers are of that era in design. The big plus was we required masks, and though there were a few grumpy students to start with, most did as students do and simply adjusted. We have had our share of cases, but have remained remarkably low, I feel in large part to our staff and students keeping masks on and staying as apart as we could. Masking, I truly feel, saved us. Cases in Wisconsin spiked early, and in this provided link, you'll see a correlation to our state being inside, and particularly when schools almost all reopened September 1st. I feel like someone could get their PhD studying our state. Our Supreme Court ruled out almost all mitigation in mask mandate, as shown on the chart as well, August 2020. Our cases peaked before Thanksgiving and our traditional 9-day deer hunting, where tens of thousands gather at deer camps. We continued to fall through Christmas, and although there is a reporting bump right at Christmas, it nicely fills the drop that also exists during that same time frame. I really believe we ran out of persons that were out and around who could get it. My own words, quote, we ran out of idiots. Bars and restaurants have been full almost from May 2020 on, but once we went inside in September, the cases finally reflected our behavior. Our numbers are almost certainly underreported, as I know dozens of recently graduated students who experienced the classic symptoms and signs of COVID, yet never were tested so as not to lose jobs or be quarantined from college. Many others in the middle-age group also avoided testing so as not to affect jobs. I feel strongly we are at least a factor of 2 off of the 550,000 plus cases. Wow. So assuming the possibility of 1 million cases and the 1.1 million first vaccine shot folks we now have, could we really be approaching a tipping point? Our case numbers are about 450 per day and percent positive at 2%. We only have 4.7 million persons 16 and older, so maybe 2 to 2.25 million with some measure of protection. We are vaccinating 250 to 300,000 per week. So mid-April, 3 to 3.5 million with some measure, 70 to 75%. Just curious as to what you might think, and maybe a pitch to all those out there that hope is on the horizon. Here I will include some screenshots from the Milwaukee Journal Sentinel and a link so you can refer to what I'm looking at. And he sent the link, and I pasted in the image. Lastly, it's crazy and frightening when your county hits 2,200 cases or more per 100,000, and your neighbors are turned away from hospitals. Wisconsin paid a high toll, and many families are forever changed. I managed to get my 75-plus-year-old mother in for her shot and was fortunate to receive mine as well. Pfizer it was, and we get second doses St. Patrick's Day. So that's tomorrow. Please know that you may have wondered what the hell we as a country were doing, or if anything you as individuals or as a collective could accomplish. Your podcast, your steady voices, your wonderful way of inviting the newbies into your world provided many, including myself, with comfort and knowledge. My very sincerest thanks to you all. P.S. I teach in Hayward, and you should all check out the American Birkebeiner cross-country ski race sometime. Scott.
Hayward is a major vacation area for people who want to catch a muskellunge.
A muskellunge?
A muskellunge.
This must be a type of fish, right?
It looks like a giant pike or a barracuda. It can grow up to 70 pounds, and that's the epicenter for that sport. It's a fantastic place. I've been there. It's right on the border between Michigan and the Upper Peninsula of Michigan and Wisconsin. And instead of being a bar on every corner, there's a sports shop on every corner selling musky lures that are about that long.
And you know this why, Dixon?
Because Notre Dame has a biological station on the other side of the border.
Got it.
And I used to go up there about 3 or 4 times before I graduated. So it was a good place, very nice place.
So this Birkebeiner is the largest cross-country ski race in North America and one of the longest.
Wow.
Started in 1973.
I would agree with Scott that, as we discussed last time, I think we've underestimated a great deal the number of infections. So we're we're getting to a point now where I think we're approaching a tipping point, as Scott says.
Tipping point is a good term.
I like that.
Yes.
I mean, I'm not sure yet, but it's a reasonable hypothesis.
Feels right. It feels right.
Yeah.
We have only immunized about 11% of the population so far, though. I saw that on the NPR news 2 nights ago or something. So we have a long way to go, I think.
Yeah, of course, that's an average, right?
Yeah, that's true.
Kathy, did you paste this quote in here?
I'm sorry, I scrolled.
The Scott Galloway quote.
Oh, nice. I did, right.
Very nice.
Yes. Yeah. In fact, I sent it to Rich. He writes a blog post called No Malice, No Mercy. And the March 12th post is entitled Institutions. And a friend sent me just this one paragraph from it. So I'll go ahead and read it.
Please.
In recent decades, many Americans have conflated liberty with selfishness, adopting the notion that freedoms are self-sustaining, that liberty is a birthright that no longer requires sacrifice or collective action. In turn, this attitude has denigrated the institutions and traditions that yielded our freedoms in the first place and served as the connective tissue holding the nation together. These attitudes were societal comorbidities, and when the pandemic hit, the results were tragic. Despite having just 4% of the world's population and nearly 30% of the world's wealth, America suffered 25% of reported COVID-19 infections and 20% of its deaths.
Perfectly put.
Yes. Mm-hmm. Conflated liberty with selfishness. That jumped out at me.
Exactly.
And I love the notion of societal comorbidities.
Yeah.
Yep. It's good. It's really good.
Great use of English.
All right, Rich.
Adam writes, greetings, Dr. Racaniello. Not sure, but I thought you might find this interesting. I am in the Japanese phase 3 trials.
Janssen.
Hansen.
How did I do that? Sorry. I'm in the Janssen phase 3 trials, and I just talked to the people there. They are moving everyone's next in-person appointment earlier, starting first with those over 60 years of age. After the blood draw, they are going to unblind everyone and offer the vaccine to those who have received the placebo. I had previously thought that they would do the blinded crossover method, but I guess they got what they wanted from this particular trial. Thank you so much for TWiV. I discovered it several months ago and now consider it an essential part of my news and general information consumption. Plus, it's just plain fun. I'm gradually exploring the back catalog as well as other channels on Microbe TV. I can't thank all of you enough, and keep up the good work. Sincerely, Adam.
I think didn't Matt say that he thought that the phase 3 results would come out in April? I think so. I think so.
Right.
Sounds like they're reaching the end, which is good.
Yep, good.
Very good. All right, it's time for some picks. Dixon.
Yes, sir. Well, I decided to keep it light this time. I went and typed out science videos, which is an amazing collection of gee whiz, Mr. Wizard type of demonstrations of science in various kinds. And these are 10 amazing tricks that you can do with liquids fun for the whole family. But the one I was completely impressed with was the little glass vial, the circular glass vial, the magic marker pen that was black, so they made a black rectangle, and paper. And with that, if you go into the woods, you can start a fire as long as there's sunlight. And it shows you how to do that. And I think that's an amazing survival device. But it has lots of other things too. You know, the refractive index of glass and glycerin, the peaceful use of corn syrup, the way in which centrifugal and centripetal forces work for shear forces in liquids. When you insert a dye, like it looked like he was loading a gel when he actually did that. He took 3 different colors of, I guess it was corn syrup that he did it with, and he injected it down into a thing, and then he rotated it, and then he rotated it back, and the colors reversed.
Yeah, I thought that was the coolest one. I really liked that.
I had so much fun with it. I couldn't stop looking at it.
Cool. I forwarded this one to the grandkids. I thought
oh, absolutely.
Like
yeah, that's perfect for them.
Brianne, what do you have for us?
So I have an article from the New York Times that talks about a recent paper that was published on C. elegans. The New York Times article is called "How Do Blind Worms See the Color Blue?" And it's a really fascinating article where this investigator was looking at how C. elegans were able to get away from or avoid Pseudomonas organisms that make a toxin that is a problem for C. elegans. And so he was thinking about different ways that the C. elegans could avoid Pseudomonas. And one thing he thought of was, well, maybe they can tell it's blue. And of course, they don't have eyes. But so he thought that was a little bit of a silly hypothesis, but he decided to check it anyway. And so he did things like make E. coli, which are the worm's food source, blue. And suddenly the worms avoided the E. coli, and he removed the blue color from the Pseudomonas, and the worms didn't avoid the Pseudomonas. And he did all sorts of things to actually show that the worms were responding to color. He looked at some genes that were involved in this, and they didn't seem to have any known way that they could be involved in kind of sensing this blue. And it's just a really interesting story that's fascinating and makes you think about biology and makes you think about how you do an experiment.
That's nice.
Cool.
Love it. Like it. Just great. Make the worms blue.
I love it.
And I knew Dixon would also like it.
You like that,
Oh,
Dixon?
I did. I loved it. In fact, I could even think of the following joke that involves 2 worms that walk into a bar, and the bartender looks at both of the worms, and one worm looks happy, the other one looks not so happy. Says, "What's wrong with him?" He says, "Oh, he's just feeling blue."
Yep. Dixon,
That's
why the
for
long
your grandchildren also.
Dixon, why the long face?
Exactly. Well, that was a different joke. No, a horse walks into a bar, and the bartender says, "Al, what's wrong? Why the long face?"
Okay. Rich, what do you have for us?
This came to me this morning from my son-in-law, and it's an article from the local NPR station entitled "A Statewide Study Shows Evidence of Significant COVID-19 Antibodies in Children," reminiscent of the discussion that we had, last Friday, and I think some recent previous discussions as well. This study has no data in it, or the article has no data in it, no links to any data. I hope there will be a publication sometime down the road. It does link to the study itself, which is called Texas Cares, and there's a link to that in the article that shows how the study is done and how one can sign up. But the bottom line is that it's a sero survey of thousands of people in Texas that comes to the conclusion that the seropositivity in Texas at this point is somewhere between 14 and 24%, but importantly, 30% of children ages 5 to 19 had antibodies, reinforcing the notion that, yes, indeed, children do get infected with this, and at a rate that's probably similar to what adults experience.
Kids are not all right.
Kids are not all right.
Kathy, what do you have for us?
It's just a short little thing, very short little video on this site from the NASA camera aboard the Deep Space Climate Observatory, which looks like you pronounce it Discover when you put all those letters together. And it shows the moon crossing in front of the Earth, which is the Earth is rotating, and you see the dark side of the moon. And you click on it and it just goes over and over and over, and it's really fun to watch.
Cool.
So
That's very cool. I forwarded that one to the grandkids as well.
Yeah, there's no man in the moon on the dark side.
Well, you should probably play Pink Floyd while you're doing that.
Right.
This is another one of these telescopes that they have situated in a Lagrange point.
Yes.
Okay. Which just blows my mind. Way the hell out there in space, essentially stationary because it's in a position with balanced gravitational,
That's right.
balanced gravitational forces. Blows my mind.
That's very cool. All right, I wanted to give a couple of picks about keyboards. I've been typing most of my day on keyboards since the 80s, I guess. And I think it's really important to have a good keyboard and a fun keyboard since you're staring at it. I didn't always feel this way, but a couple of years ago, I decided to customize them. So I have 4 keyboards. Now, my first 3, and I've put a couple pictures in here, are colorful designs that I put together. You can go to wasdkeyboards.com and, you can pick all your colors and they'll send it to you. So that's what I've done, and it's a lot of fun. But it turns out my son, Aiden, is big time into keyboards. And keyboards, man, they are a thing to customize them. There's a whole world out there. Oh, my gosh. So he turned me on to this company, novelkeys.xyz, and they're making keyboards. And mostly they're sold out because people go and buy them all out right away. But I got one a couple of weeks ago. It's just the base. So now what you do is you buy a base, which is empty. And I bought one that has an orange plastic rim around it. It's very solid and heavy. And then you buy switches, and then you buy the keycaps. And the switches come in all different kinds depending on whether you want clicky or not, or resistance. And I like clicky, but when you're podcasting, you can't have clicky. So my recent one, I tried to get quiet switches, and there's some that are quiet, some that are tactile. Oh man, you can get down a big rabbit hole with this stuff. So he advised me which switches to get. You just push them in, and there's a picture there showing you the board without switches. You just push them in, they sit right in, and then you get keycaps. And so again, you can design your own keycaps. You can buy sets. So I bought a set. I was looking through the set, and I found this called Matcha, which is different shades of green based on the tea. And I just love the colors. And I put it together, and it's great. It's nice. It's tactile, but it's quiet, so I think I can use it for podcasting. We'll see. But it comes with little keys with like an ice cream cone and a cupcake, and you could choose to put them wherever you want on the board. No, for keys, you don't use that much, I guess. What were you going to say, Kathy?
Can you get them with the really short keys, the short profile?
Oh, you can get anything. So, if you're used to the Apple keyboards like this one, right? This one I got from a colleague here who left, Saul, and this is flat, and this doesn't make a lot of noise. So this is kind of good for podcasting. But if you're doing extensive typing, I don't like that. I like to have a little tactile feedback. So I use these, but you can get flat. And keys come in all kinds of shapes. For example, the last one I got, the keys are all the same height, but some boards, they change as you go from the top to the bottom rows. And also the part where you put your finger, it can be bigger or smaller. It can fill the whole space, or in mine, you can see it's kind of a shape. It's just crazy what people are doing out there. And there are many, many other websites, and I'm sure there are many people who are listeners who are into keyboards. But,
I wonder what fraction of your brain you use for this. This is amazing.
So I just like looking at the colors, and then it feels different. Every keyboard is different. I was watching a YouTube video last night of a guy. He was talking about the different kinds, and he has on his wall all these plexiglass shelves, and he keeps his keyboards, and I guess he just pulls one out and uses it. different day, and they have all different designs and colors and keys. This is a thing, folks. It's amazing.
Wow.
So anyway, I thought I would give a little homage to keyboards. I really like keyboards. They're cool. All right, we have a listener pick from Bronwyn. Dear Team TWiV, greetings from Sydney, Australia, where it's a mild autumn day, currently 20 Celsius, 68 F. I am one of the many non-scientists who found your podcast during COVID. Your reasoned collegial voices have been the soundtrack to my days in what has been, even in Australia, with relatively low numbers, a challenging year. So much so that when I heard the news of the Texas freeze, my first thought was for Rich and his family. Following Dixon and Rich's interest in the recent New York Times articles on different countries and COVID, I thought you might be interested in this website from an Australian think tank, which allows you to compare different countries on different factors. At January 2021, so some things have changed since. You might wonder why an Australian think tank would be prioritizing this. It's possibly relevant to note that as a country with a relatively small population, 26 million, Australians tend to be fixated on our place in the world and really annoyed that our neighbor New Zealand has beat us in the COVID stakes. And he gives a link for that. Or she, not sure. COVID Performance Lowy Institute categorizing countries based on their population side revealed the greatest differences in experiences with COVID-19 challenge. These results stand even after taking into account per capita indicators to evaluate performance, minimizing the likelihood of a methodological bias against countries with more infections because they have larger populations. Thanks so much for your work. I'm proud to be a patron and I'm appreciating your back catalog, which may serve as a great resource when someone comes to write the definitive history of COVID. Maybe a COVID version of Randy Schilt's classic And the Band Played On in relation to HIV, not to mention all the other fascinating and scary virus stuff. Best wishes and stay safe. Bronwyn from Sydney. Thank you, Bronwyn.
So this website has this company has a cooked up COVID performance index that I didn't look into exactly how they did that, but it's basically their measure of how well different populations responded to the pandemic, and then they correlate that with different other variables such as the population, the economic prosperity, and et cetera. And as already said in the letter, the strongest correlation between how well you did against the pandemic using their measure and anything else was population size. Countries with larger populations had more trouble than countries with smaller populations, which makes sense.
Yeah, it's interesting how it changes over time too. Very cool.
It also catalogs that developed advanced countries did better than developing countries, and et cetera.
It's really cool too, because so the right-hand side is the score, and on each graph, toward the top is better and toward the bottom is worse. But on the left-hand side, there's different tabs, and you can click on them. So you can click on this, the first one, you can click on the Americas, and then you'll see all the individual countries in the Americas. And being toward the bottom is worse. And at least in the first two graphs I looked at, the United States was worse. And then there's one that's based on political systems that was interesting. So there's a lot in here when you start clicking through.
Yeah,
it's true. So the Times had a follow-up on the graph that I sent in, and they had another think tank moment, and they published another analysis that showed there was a direct correlation between obesity and number of deaths. And the United States is the heaviest nation in the world.
Yep.
So that may explain a lot of things.
Sounds like the one that Rich picked, maybe?
Yeah, that's what I picked that follow-up for you last episode, Dick.
Okay, good.
And also bounced it off of Stacy Schultz-Cherry, who's our obesity / vaccination / infection expert, and she thought that was a fine correlation.
Yeah.
All right. That is TWiV 732. Show notes, microbe.tv/TWiV. Send your questions and comments to TWiV@microbe.tv. Now, if you want to send a question to Daniel, that's Daniel@microbe.tv. And if you like what we do, consider supporting us, microbe.tv/contribute. Dixon DePalmier is at Racaniello.org, thelivingriver.com. Thank you, Dixon.
You're welcome, Vincent, and everybody else, and have a happy St. Patrick's Day tomorrow.
Oh, that's right.
Your shirt's one day early, but it's nice.
Oh, I didn't think of that. I just grabbed it.
Well, there's some subconsciousness about all of this. Perhaps it crept in.
Yeah, there's a big corned beef in the fridge, and I said,
"Yeah,
to
we got
Doris,
ours. We've got
I said,
ours.
what is
We've got
this?
ours."
What is this big slab of meat?" And she said, St.
Exactly.
Patty's Day. Oh, that's right. She makes it every year.
Yeah, we do too.
I'm not a big fan of potatoes and corn. I'm sorry. It's just not very interesting.
Actually,
It's very Italian, I must say.
sounds really good to me. I haven't had that meal.
Yeah,
in a
that
long
sounds pretty
time.
good.
The best
we
thing-
have it once a year.
The best thing is pasta. I made rigatoni with sausage on Sunday. Oh my God.
You see.
Oh my God, it was so good. I put a couple of cans of tomatoes, you
Listen to
saute.
him.
Holy cow, there's nothing. And a little grated Parmesan on this. When I grew
Forget
up,
the
we
red
used to
wine.
have pasta on Sundays. And so
Really?
now, if you ask me what's for dinner on a Sunday, pasta for sure.
How interesting.
Just because it's a habit, no other reason.
Yeah, but you got used to it, though. That's good.
Brianne Barker's at Drew University, BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here. Now you guys are making me hungry.
Rich Condit's an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
And Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
I'm Vincent Racaniello. By the way, Kathy, thank you for organizing the Rohwer and Rojas sampling study.
Oh, and earlier I found,
very good.
I think it was TWiV 617 where we read
Okay, cool.
Marisa. Marisa is her shortened nickname.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back in a few days. Another TWiV is viral.